Thank you very much for joining this session. Thank you very much, Lance. My name is Andrés Caicedo. I'm professor and researcher at the Universidad San Francisco de Quito in Ecuador. And I'm also scientific advisor of several companies and initiatives. And at WGAs, I'm the team leader of Dragon Biomed, which is a USFQ initiative aiming to translate all that we do in the lab to close to the patient. I'm also the CSO of Omnios, a well-known biomarker company, and also CSO of Orbital Foods, redefining cuisine for extreme environments. So, as the field is, well, very new, we still receive a lot of these questions, like what is mitochondrial transfer all about? And we felt some time like this character, in which we presented several of our science, great articles made by Jonathan, made by different people here, and however, there are still a lot of skepticisms, and they still think that it's quite like an artifact. But the good thing, and that's why we are here, is because no, there are evidence of the transfer, and there's also evidence of the therapeutic potential. So I would like to thank Lance, James, and Melanie for this amazing opportunity to bring together scientists, companies, and of course patients. So this is super important. So a few years ago in Chile, we get all together with Jonathan, with Keshav, and many other great scientists, and we developed this amazing paper, which is recommendation for mitochondrial transfer and transplantation, nomenclature and characterization. And we, gathering all the evidence, observed that there are two main mechanisms of natural transfer of mitochondria. There's one that is content-dependent, and the other one is, of course, content-independent mitochondrial transfer. And today there are many different lines of evidence in which we can see that the mitochondria is, different, and it uses these different mechanisms, and these different mechanisms have a lot of connotations, whether it's in health or in disease. So talking about content-dependent mitochondrial transfer, it's been observed and reviewed by many great scientists, for example, Jerry Nussel. Here we know that when tuning nanotubes are developed, he postulates that this process is happening a lot when cells are under stress. And when cells are under stress, they build these nanocellular connections made of tubulin of actin, and they use myro as to transport mitochondria. It has been observed if you overexpress myro, there are more transfer of this organelle from the donor to the receptor cell. Then we have these two very interesting aspects of tuning nanotubes and mitochondria. What we know is that, for example, you have kind of like good mitochondria that is transferred from mesenchymal stem cells to CD8 plus cells. And when CD8 plus cells receive this transferred mitochondria by not only myro but with TL2 protein, they overcharge themselves. It's like they do and perform better fighting the tumor. So in this context, mesenchymal stem cells that have good mitochondria transfer it to CD8 plus cells, and they do good. But in other contexts, you have tumor cells that transfer mitochondria to tumor-infiltrating lymphocytes, and these cancer cells transmit mutations, and also these mitochondria seems that they are in bad conditions, and they induce senescence and metabolic problems in the cells that are receiving them. Actually, the immune cells. So cells are performing less, and they have a lesser capacity to fight the tumor. So these two aspects are very, very important. Then, in another factor related to content-dependent mitochondrial transfer, we have those that are dependent of gap junctions. So in this seminal article, Islam showed that in ARDS model, so you have a mice that has this problem, and then you administrate mesenchymal stem cells, and the mesenchymal stem cells get in touch with the alveoli. They form the gap junction, and they transmit good mitochondria. And it has been observed that they increase the quantity of ATP and also improves the surfactant production in these cells and tissue. Then there is other article, very interestingly, in which mesenchymal stem cells are able to receive mitochondria from platelets, and that energizes them, and they perform better when they are doing the wound healing. So this is very interesting, very importantly, because we've seen so far mitochondria in different contexts, in which first good mitochondria is transmitted to the good cells, and that also mitochondria can be transmitted to the wrong cells, to the bad cells. And here's what we show that, for example, mesenchymal stem cells that are supposed to have good mitochondria, they are transmitted to cancer cells, and they are improving their function, they proliferate more and demigrate more. So, so far it's important to further understand the different characteristics of the transfer and how different types of mitochondria are inducing different effects. So, and then regarding, for example, gap junction and connective C43 mediated processes, we still need to gather more information about this, because it seems that the gap junction is probably transmitting not only good mitochondria but also mitochondria that is damaged, and it is super important in order to better understand this aspect of regenerative medicine and the application of mesenchymal stem cells in transferring mitochondria to damaged tissue. And we recently, together with Dr. Stefania Nova, we made a webinar in which we highlight these key points, like mitochondrial transfer, the good side and the bad side. And it's super important to discuss that because we are at the door of developing better ways to produce mitochondria to make cells liberated better. For example, generating ways to have a good quantity of them for off-the-shelf, for example, therapies, and that is key to understand this part. Then we have content-independent mitochondrial transfer, in which we have free mitos, is like mitochondria completely naked, devoid of any membrane structures, and then we have those that are enveloped in microvesicles. And in this article, Marco Rosina observed that, for example, fat tissue, brown tissue, that help us keep us warm, they use a lot of mitochondria, but they have to get rid of it, and when they get rid of it, there come macrophages that take this harm mitochondria, and they clear it out. Because if they are not there, and if you block this mitochondria that is enveloped in microvesicles, the adipose tissue just starts to stress more. So this is something very important, and it has been observed that if you have mitochondria that is enveloped, they could be damaged, so they need to be cleared out by the macrophages. So this is a very important aspect. And then we have cardiomyocytes that, you know, they use a lot of mitochondria. So when their mitochondria is not working well, they have to get rid of it, and we have to have macrophages that come and they clear it out. Because if there are not macrophages that actively clear it out by, for example, mechanisms using more TK, for example, instead of heparan sulfate, they accumulate in the cardiac tissue, and then they start to do harm. So it is, in this context, it's super important to understand that the different packages of mitochondria could have good or bad. Then we have this great article made by Jonathan, in which we have other aspect of the mitochondrial transfer. So we have adipocytes that get rid of their mitochondria. There comes the macrophages again, but if you change your diet, you have macrophages that doesn't take this mitochondria, and then you have circulated mitochondria that actually are going to the heart. So in one particular aspect, you have cardiomyocytes that need to get rid of damaged mitochondria, and then you have the adipocyte mitochondria that start to circulate, but then it goes to the heart, and it seems that it's doing good. So just is making the field a little bit like complex to understand, because we need to know which one is the best mitochondrial source in order to use it for therapy, for example. So mitochondrial independent contact transfer is very complex. We have different types of mitoses that are embedded or not embedded, like free mitoses or ED mitoses. And the presence of these free mitoses or associated mitochondria does not necessarily indicate whether they are healthy or damaged, so we need to understand this far more. And the uptake of ED mitose that sometimes are damaged seems to be mediated by CD36, and when there is free mitose, there seems that we need, for example, heparin sulfate or MERTK receptors. So recently, in an article by The Economist, this is part of the great expectations that the field has, recently published that mitochondrial transplant could cure diseases and lengthen lives, but we still don't know a lot of things. For example, is horizontal mitochondrial transfer always a good thing? Which kind of patient could receive it? And many other aspects that we need to further study. So regarding artificial mitochondrial transplant, we have the isolation. So we use different types of methods. We have kits. For example, I use thermoscientific kit. Jonathan uses the Milteni one based on beads, and we have, of course, the one that uses James, which is something that he developed in his own lab. But maybe the mitochondria that we are isolating is changing accordingly to the different kits that we are using, and probably the effects that we are expecting or seeing is dependable of the process. Melanie showed in her paper that there are key aspects that are super important, and I think that we all agree on, is that we need to characterize the mitochondria that we are isolating, checking if their morphology is okay, because we know that CRISPR tell us a lot about the mitochondrial function and the morphology, and we always focus on minimal quantities of contamination. However, maybe the method could be bringing something extra together with the mitochondria, and that could be part also of the beneficial or damage effects that we will see. So this is something that we should take care in consideration. And functional testing of the mitochondria is also imperative. Then the mitochondria isolates are studying stability and preservation. So James mentioned that, for example, if we isolate mitochondria, they probably last for hours. But we all know that it is important to preserve it, and there's something that we've been working on is a mitochondrial preserving agent, and how we assess if this mitochondrial preserving agent is good or bad is by transferring this mitochondria to the recipient cell and checking if there are changes in proliferation or MTT activity of the recipient cell. So far we've seen that the preserving agent is working well. The mitochondria has been preserved for 72 hours at -20. So then there's the administration, culture cells receive well mitochondria. We have transferred mitochondria to 2D supports, also cells that are not attached. Then we have the autologous transfer of mitochondria coming from muscle biopsies to the heart, and then you have different forms of isolated from, for example, from PVCs or placenta, and used even allogenically. So what happens in the culture cells? We have an increase of mitochondria, and it's also we will note that the transfer of mitochondria induces changes in the recipient cell, induces biogenesis, and you have more mitochondria, but it also increases their respiration. So this is good. And then we have the ex vivo transfer of mitochondria to the recipient cells, and then use these cells in order to transfer to patients. And this is something that I'm really eager to hear about in the presentations of Noah and Conviable. So I think that we want to speak a lot about this process, which I think is more specific and maybe a way to treat patients better. Then systemic administration of mitochondria is quite complex. We have observed recently that you have extracellular mitochondria in the blood by the work of Dr. Alan Terry. However, if you infuse mitochondria, you could expect that a lot of cells will take this mitochondria in different patterns, as shown, for example, by John. You have a particular tissue, and there is extracellular mitochondria. Many different types of cells are uptaking it. in different patterns. So this is complicated because it makes the system more interesting to study. And we have observed already if you transfer mitochondria to pillion Cs, you have different patterns. For example, you have CD3 and CD14 plus cells that actually take mitochondria differently. So if you administrate it, there will be like different cells, and it will be challenging if you are doing it for a patient. So one other thing is that we need to think about is like if you administrate, for example, intravenously, you could have good cells or bad cells that are going to take that mitochondria. So it is important to really know the profile of the patient. And the administration should be, for example, if you administrate mitochondria, so far it seems that if you use the in situ administration is better, as shown by Dr. Gianlucoli, and also in this article that was made by Dr. Pachilus in Chile, the administration of mitochondria in OA, in a normal arthritis model, seems that it's working and is decreasing the OA score of the tissue. And recently we are working in transferring mitochondria to the wound tissue and observing what's happening there. It seems that it has good effects in inducing the regeneration of the skin. So it seems that extracellular mitochondria isolated and infused into cells induces different mechanisms of activity. In this case, it seems that induces micropinocytosis, which could be different from the one that is actually secreted by the cells. So together with Dr. Stephanie Noma, we would like to work on this in order to understand how different is mitochondria that is being isolated because you are taking it out from the cell, and the mitochondria that is being produced by the cell and is liberated. So we would like to do proteomic and functional analysis. So if you would like to understand this, we should try to speak a little bit. And then, if I mentioned to you that mitochondrial function is important, we did all the other way around. We stress the cell with UVR radiation. So we bombard the cell with the UVR radiation, and then we check the correct dose in order that we could actually isolate these mitochondria, not too damaged. And then we put it into cancer cells and then in normal cells. So we observed that cancer cells and normal cells uptake these mitochondria, but actually these harmed mitochondria perform differently. Here at the left we have the cancer cell that took the mitochondria. So here you can see that the damaged mitochondria actually decrease the proliferative potential of the cancer cell. And then in this part we have the mitochondria that actually perform good. Then we infuse this damaged mitochondria in the tumor and induce a massively attack of immune cells, as you can see here, and decrease the size of the tumor. So we are looking forward to collaborate and check this thing out. And we need to check the type of mitochondria, the type of transfer, in order to move forward to the patient. And thank you very much for everybody for being here and eager to answer any questions or do collaboration.